And now for the section 4, the part of germplasm, we will have the invited talk of Dr. TERZIC. Dr. TERZIC works as a breeder for germplasm enhancement at the Institute of Field and Vegetable Crops here in Novi Sad. He leads a program on plant genetic resources and pre-breeding research at the sunflower department of this institute. The main focus of his research is the use of wild sunflower relatives for the improvement of the cultivated sunflower. This is done through a non-stop process of evaluation of the germplasm and introgression breeding with the valuable traits that are discovered in the process. He has evaluated the wild species of Elianthus for traits like disease resistance, for broom rape resistance, or for tolerance to or resistance to herbicides. And he has identified promising germplasm in the wild species. He has created specific combinations. And finally, he has used the interspecific combinations for the breeding programs. Today he will talk about the importance of the genetic resources in sunflower breeding in all the phases, from the conservation to the introgression breeding. Dr. TERZIC, thank you.
Thank you very much for a very nice introduction. It's an extremely big pleasure for me to speak to you today. I feel very strange probably because I was so much into organizing the conference, so I'm just a part of the system. I don't have maybe the nervousness I should have about the presentation, but I just feel comfortable, and thank you for being here, all of you, today. As announced, the subject are genetic resources. I will comment a bit on the wider scale internationally, but focusing more on our experience from Serbia. Again, a bit specific environment for work, but we, I think, managed to accomplish a lot. Some speakers before me mentioned about history. I will also go a little bit behind. So, from the 19th century, the development was quite fast, and sunflower reached a very high position, number 2 on the oil seeds, after soybeans, and the next big step was the production of hybrids. So we turned from, this is also Peredovic on the upper picture, turned to a factory-like production. So plenty of times we, as I said, revisited ancestors, the crop wild relatives. Plenty of people now famous have done that, and I will only mention some. You recognize the names, and they all contributed greatly to what we have in the fields now. So in 1979, an FAO sub-network was established, and the institute in Novi Sad at that time was announced as a communication center. We are also celebrating a nice jubilee. It's 60 years from the foundation of, then it was called the Industrial Crops Department, now it's Sunflower Department, but that is how long we are working on sunflowers, officially, even before that. I myself am celebrating these days 20 years of work at the Institute. And in 1978, we were among the first to produce sunflower hybrids on the market. And the collection itself, which I believe most of you will see on Thursday, was established. So when we speak about pre-breeding, the process of preparing exotic germplasm for the use of improvement of cultivated species, not only for sunflowers, it's similar to other species. There are some usual steps with collecting germplasm, characterization, selection of optimal accessions, and at the end crosses and crop improvement. What is important, I will also mention it on some other slides, it's a time-consuming process, and if everything goes well, it takes about 15 to 20 years to do it. That's why the name of the section is Investment for the Future. So these are some steps that I will go through in this presentation. So the material is, of course, the Elianthus genus. plenty of sections, series, and species. Some easily crossed, some not so easily. I think the only one is Helianthus porteri that has not been crossed, maybe in the meantime. And of course, it's not a job that a single institution can do. Plenty of institutions supported the efforts, based on, of course, the USDA, as the center of origin is America, and many like FAO, ECPGR, Scorena, USAID, and other participated. About the people from Novi Sad, most of the expeditions were made in the 1980s, and not only in the US, of course in Canada and in Montenegro. There was also one very nice expedition of collection. Before the 1980s and in the subsequent years, we also exchanged materials with other institutions from France, Bulgaria, Romania, Argentina, and other countries. And of course established cooperation, which was very fruitful. After successful collection, if that is the desire, there comes maintenance, which is not so easy as it seems. Even though they are wild species, they do originate from some specific environments, and we lost some accessions due to the fact that they couldn't, for example, finish the vegetation cycle. They flowered quite late, and the frost comes in Novi Sad even in October, November, so they just couldn't produce seeds. So this is how it looked like in 1984. We had a sort of dense speculations in 2008, and now it is something like this. We use a simple weed control fabric to reduce the number of sunflower plants, but also to keep the weeds out, to reduce the number of field hands that must be engaged in maintenance. This is a short list of annual species, 7 of them. The first column in green is the total number that entered our collection, but with seed reserves, you can see we have 74 Helianthus annuus, 25 petiolaris, and so on. And due to the self-infertility or self-incompatibility, the number of seeds per accession is very variable. Similar for perennial species. We have them also available with seed reserves, but most are kept as a living collection, which we will see on Thursday, with different number of accessions per species, most of them from Helianthus tuberosus. We use two types of isolation to keep the genetic identity of accessions, cage isolation or paper bags. And apparently cage isolation is a bit more demanding, but provides much higher quantities of seeds per single regeneration. We usually do it by hand, but we also used insects, and we had the most success with bumblebees. This is a commercially available hive, very small, with 15 working bees, and they were quite enough for the small cages we use. So when you conserve seeds in gene banks, it's a matter of storage conditions. We use medium storage at +4 degrees, and in those conditions we usually the seeds lose germination below 50% after some 6 to 7 years. This is for Helianthus debilis, and then between 5 or 6 years we just do another regeneration to increase the quantity of available seeds. The method by Gentler and Jen was mentioned. We also use it to increase the germination by seed scarring. When we do interspecific crosses, we also do them with between cultivated sunflower and perennial species. And in F1 generation, those plants keep the ability to produce tubers. So we are not obliged to make further back crosses in that year. We do have them still in the collection, and some of them even for several decades now. So they are available, fixed in that F1 stage for further research, back crossing, etc. Now, maybe the most important step is the characterization, without which the germplasm is practically useless in gene banks, because only after this step we know which to select according to the desires of breeders. And not only of sunflower, more than close to 60% of traits is about pest and disease resistance. Okay, so this is we also have now it was the first publicly available association mapping population from the University of Georgia. We evaluated some morphological traits and found, for example, the largest diversity in the upper left part. Doing something wrong. And the lower left are the oil lines, fertile lines, and on the right are the restorer lines. These are tubers for Helianthus tuberosus. We also use some of the species as they are, without process with cultivated sunflower, and even registered 2 cultivars after evaluation of some desired traits. About new traits, we have a very good cooperation with the Faculty of Biology in Novi Sad, with a very young and inspiring team, and they contributed much to finding potential new traits for improvement of cultivated sunflower. And if you look at all the species that are used as crop wild relatives, according to Corey from 2015, Helianthus can be declared as a model genus for reproduction breeding, because the largest number of species is used from that genus, mostly for biotic stress resistance, but also for cytoplasmatic male sterility and fertility restoration genes. To accumulate data in our database about resistance, we also noted the spontaneous occurrence of diseases, just to ease our work when we select accessions for a specific trait. So this is a list of perennial species that are in our collection, and even though not a large number, but powdery mildew was found with a low aggressiveness, and only Alternaria and a few cases of stem canker and Sclerotinia head rot. In annual species, a higher number of diseases, which is unfortunate because they are all closer genetically to cultivated sunflower, but unfortunately more susceptible to different pathogens. Orobanche was mentioned. It is one of the major programs at our institute also. The idea is, when available, to use cultivated lines, and that's what we call a short-term program, but we also do in parallel long-term, directly testing the wild relatives and then doing the crosses interspecific, testing F1 hybrids, and doing the subsequent back-crossing and testing. We also tested the complete collection of annual species, hoping to find some genetically more close to cultivated with resistance, and we found some good sources with Helianthus petiolaris and Helianthus niveus. About other diseases, we also have worked with the mentioned white head rot and macropomina. And the sources were found in the Helianthus debilis and Ergophilus, and for macropomina, Helianthus annus, tuberosus, and petiolaris. Now, for dummy mildew, it is known that resistance can be found both in annual and perennial. But as our colleague Josip said, we should keep in mind to select the accessions that are the closest to the cultivated sunflower, not to disrupt too much recombination rates and the required traits of cultivated lines. For FOMA, it is also tested in controlled conditions. And for Phomopsis, I think it's now already famous, the sources that were found and created at our institute. They are still, after a few decades, quite resistant, even to current occurrence of that pathogen. It is not only biotic, we also have tested with express total herbicide and found good resistance in several accessions of Helianthus ergophilus, and that material is currently also in work. Now, why biotechnology separately? Because exactly natural hybridization is possible in the genus Helianthus, almost between all species, and it occurs even spontaneously. So it's a bit difficult sometimes to assess what are the potential difficulties and potentials. So that's why we use biotechnology to assess the difference, starting with chromosome number, then chromosome structure, which all affect problems that occur in crosses. But as mentioned by Jen, solutions fortunately are available. So if the cross incompatibility doesn't occur pre-zygotic, I mean before the pollen germination, we can have normal stages of meiosis, but usually it is not so. So these are the pictures of chromosomes. Usually it is more like this. Plenty of multivalence and fast or lagging chromosomes that all contribute to this, that in parent lines, wild and cultivated, the pollen viability is close to 100%, but in F1s it's becoming very fast quite low, and that complicates further backcrossing. So I have only one slide exclusively on genotyping because it's not my field of work. There are other speakers that will speak about it in more detail. But some major advances were made. Today we have several genomes sequenced, and what's even more important, we have the International Consortium on Sunflower Genomics, which even announced sequencing of reference genomes for wild sunflowers and wild relatives. So I think it's a very good, something very good in the future. For the crosses themselves, this is what I mentioned about unwanted traits. This is a schematic of Helianthus salicicolis, one of the most diverse or more distant by phenotype. But most of them can be crossed and are used. It's just a matter of an opinion which to select to bring the wanted traits. The success rate is the highest with, as you might expect, Helianthus annuus, followed by Ergophilus and Petiolaris with annuals, and in perennials it gets difficult very quickly, and there we employ techniques like embryo rescue and chromosome doubling, etc. So this is for some of you who haven't had a chance to see. The wild parent is Helianthus rigidus in this case. The cultivated line is what you're used to see. And the F1 is in the middle, having most of the phenotype traits inherited intermediary. So, as Randolph nicely reviewed in this paper, not only for sunflower, but also wheat, potato, peanut, and other species, large majority of traits are for biotic stress, followed by male fertility, abiotic stress, and others. So, how can we help? High throughput phenotyping is often mentioned. It's a very popular nowadays. But in wild species, one small but important fact is that accessions are populations, not inbred lines. So they are not homogeneous, and that has to be taken in account when doing phenotyping. So species are diverse, also in vegetation phases, habitus, making automated phenotyping a bit more demanding. But we have, as I said, greenhouse platforms, aerial or ground-based vehicles that help that process. We create a lot of information, but due to the fact that those new techniques are quite financially demanding, we often have some limits in information sharing, and sometimes there are other reasons for low information sharing, and that I think can be improved. Eurisco is a European search catalog for plant genetic resources that also can have characterization data associated to the accession. Then AGES is an even improvement of Eurisco. It's not new, but it hasn't reached the imagined stage of development yet. And the goal is to have a proven quality of data associated, and that the accessions are really available. The US National Plant Germplasm system is probably still the most used and the richest source of information and accessions about Helianthus. And the French, maintained by INRA, the PhenoDB database, is a good example of how that can be organized and done. Now some real-life examples. Helianthus mollis with sessile leaves was mentioned already, with the idea to produce sunflower plants that occupy less space and therefore increase the number of plants per hectare, and so to have a larger yield. So all the necessary steps were done, but unfortunately the inflorescences were not inclining. They were directed toward the sun, so they unfortunately couldn't be used, but maybe in some new try it could be successful. For bloom rate, we did, as I mentioned, all the first steps. We tested annuals and perennials. We continued with annuals just because of their closer genetic identity to cultivated sunflower, and are now working on the development of specific lines. For Sparrozinia, we have something that is not uncommon both in companies and in institutes, faculties. We have permanent intraspecific gene pools, from which we selected, for this case, 44 progenies with annual species, from annual species, did the field tests, etc., and found that material from Trillium steadily had the highest tolerance. About confectionery, which was just described, this is now exactly what I showed at the start about schematics for pre-breeding, the time needed for some trait introduction. So here it's both diversity enrichment, but also some specific trait integration. So we used lines obtained from the USDA that are publicly available. It has taken 6 to 7 years for pre-breeding, then 3 years of selfing, field observations and pedigree selection, and then another 8 years to produce CMS form. So if everything goes well, then it's about close to 20 years to produce the lines. Here we have, again, oil sunflower. This material developed at our institute. Line HA26 was crossed with the listed annual accessions. And again, if you get together all the needed years for B lines, for CMS, it's again close to 20 years. So what about future prospects? I would say that phenomics will certainly help, in some cases by itself, crop-wide relative exploitation for introgression breeding, just by increasing the knowledge about them and knowing their diversity. I mentioned ages. I think that the mutual cooperation between gene banks should be increased, and to tackle some issues like even gene bank standards, just to check are we doing the right thing, germplasm availability, characterization data, but also genetic drift and pollination methods in some cases. So intellectual property is something that we are all very dependent on, especially breeders, and it's important to protect intellectual property. But sometimes it also limits a bit cooperation and information exchange. So it needs maybe some fine-tuning if possible. So, but cooperation is of course necessary, especially for international and large issues like Rocinea, broom rape, and other. I want to give a special thanks for a group of people where I was very grateful to be a part of. A team who published a paper which you have in the OCL number, thematic issue, which you have in your material. It's about gene banks for wild and cultivated sunflower genetic resources, which shows in a few pages what wealth of material and information we have available. And as Jen nicely described, it is all there for us. It is up to us to really use it. Last but not least, thank you to all my colleagues who are working now at the Institute and who contributed also to my career. I hope I at least a little contributed to theirs. And thank you for your attention with this nice slide of material used originating from the wild species.